Hello everyone and welcome back to our channel. So as you are all aware of, we were discussing about the overview of the renal physiology in the earlier videos, where we had discussed about the various processes which are taking place within the nephron. So there are three main processes which aid in the renal physiology, that is the functions of the kidney. That the main function is the urine formation. So this particular function of the kidneys, it is made possible because of these three important processes, that is the glomerular filtration, tubular secretion and tubular reabsorption. Okay, so we have seen how filtration takes place, what is a membrane, what are the different factors which are responsible for this alteration in the GFR. We have seen what is GFR. Also how the reabsorption and secretion takes place, what are the different transporters which are located in different parts of the nephron. Okay, so with this overview of the renal physiology, we shall discuss the physiology of urine formation in this particular video. So you need to have the basic idea about the renal physiology. What are the important processes which are responsible for this urine formation? Okay, so if you have the background of the renal physiology, it will be easier for you to understand the physiology of the urine formation, how exactly the urine formation is taking place in the kidneys. Okay, now as we talk about the urine formation, now usually this process of urine formation, okay, it varies depend upon the homeostasis of the body. Okay, now what is the main aim of this production of the urine? It's to remove the waste out of the body, right? So depending upon the fluid intake, okay, the urine formation is altered. Say for example, your fluid intake, it can be highly variable, right? Is it that you drink the same amount of water every day, okay, or the fluid consumption may it be in the form of water or some other liquids, or the fluids which are present in the food, does it remain the same every day? No, it alters. Even season wise also it alters. But what about the total volume of the fluids which is present in the body? Does it change? No, it normally remains stable. Okay, now what makes this fluid volume to remain stable? Why it does not get altered? Is because of the homeostasis, okay, which is maintained by the kidneys. So according to the requirements of the body, the kidneys are able to regulate the rate of water loss in the urine. So the amount of water which is lost in the urine, that can be altered depending upon the situation or depending upon the requirements of the body. Okay. Now normally the kidneys, they produce a large volume of dilute urine when the fluid intake is high. Supposing if you consume a lot of water, the urine output also increases, right? The volume of urine which is produced, that increases. And the urine which is produced, it is dilute urine. This is very important. Now why a dilute urine is produced? What do you mean by dilute? Yes, means something which is less concentrated. Okay, so urine, it comprises of a number of solutes which are dissolved in the blood, right? Various ions are present which are excreted in the urine, or certain drugs or some waste material will also be excreted out. So these will be present in which medium? Obviously it will be present in water. So water will be the solvent over here, into which several solutes will be remain in the dissolved form, right? Whatever is suspended, like your proteins, large proteins or the blood cells, these are not excreted in the urine, right? Okay, so this urine which is formed when your fluid intake is high, it is dilute, means what? The amount of solute, it is less, and the amount of water will be more. Okay, so any solution, this will be as good as a solution, right, which will contain several solutes which are dissolved in the water as a medium. So this particular urine, it will be dilute in nature. That means there will be less amount of solutes and more amount of water will be present in this type of urine. Now when it is excreted? When you consume a lot of fluids. So a large volume of dilute urine will be produced. And when will be the concentrated urine be produced? What do you mean by concentrated urine? It will be exactly opposite to this, right? What is a concentrated solution? So what happens over here? The solutes, these are more. And what about the water? In this type of urine, it will be less. Okay, so it's a concentrated urine which is formed. A small volume of concentrated urine is formed when your fluid intake is low, when you consume less amount of water, or if the fluid loss from the body is very large, say for example in case of hemorrhages or dehydration. So at that time, small volume of concentrated urine will be produced. Now what makes this possible? What makes the formation of dilute or concentrated urine possible? Why it is produced? Depending upon the situation. How it helps in maintaining the homeostasis? What makes it possible? That's mainly because of this particular hormone ADH. Now ADH, it is the hormone which is secreted by posterior pituitary gland. It is anti-diuretic hormone, right? Okay, so we have seen how this ADH, it plays a role in the renal physiology, right? So it controls mainly the loss of water. So the levels of ADH will decide whether water will be lost, whether more water will be lost or less water will be lost from the body. Why? Because there are certain cells called principal cells in the late distal tubule and the collecting ducts which are sensitive towards this ADH, right? The principal cells. Okay. So in these cells there are receptors for this ADH. So under the influence of ADH, what happens? More and more aquaporine 2 channels are inserted, right, in the tubules. So what are these responsible for? These are responsible for the reabsorption of water. So what will happen in the absence of ADH? What will happen? We have seen that these aquaporine channels, these are removed, right? So what will happen to the reabsorption of water in the body? It will be decreased. So as a result of this, more and more water will be excreted in the urine. So what will happen to the urine? A very dilute urine will be produced. But what will happen if the ADH levels are very high? So it will result in the introduction of these aquaporine 2 channels into the collecting ducts, as a result of which it will increase the reabsorption of more and more water into the blood. So as a result of this, what will happen? The amount of water which is excreted in the urine, that will be very less. So as a result of this, what will happen? Less water means what type of urine will be formed? Concentrated urine will be formed. So it's mainly the ADH which is regulating whether dilute or concentrated urine is produced to maintain the homeostasis of body fluid volumes. That's very important. Okay, so in this particular video, we'll be discussing about both the formation of dilute urine as well as formation of the concentrated urine. So coming to the formation of the dilute urine. So we'll be discussing about formation of the dilute urine first. Okay, so as you all are familiar with this particular structure. Okay, so that's the nephron which is depicted in this particular picture. So we have seen that this is renal corpuscle and these are all the tubules, right? So these are your glomerular capillaries at which the glomerular filtration will take place in the capsular space. So the filtrate will reach over here and it will travel along the tubule. So as it is traveling, we have seen that processes like tubular secretion and tubular reabsorption will take place. Okay, so as it moves into the PCT, there are different types of transporters which we have seen, right, in the PCT. Mostly the sodium reabsorption will take place over here. There is sodium glucose symporter, sodium hydrogen antiporter, bicarbonate reabsorption takes place. Apart from that, calcium, magnesium, okay, several other substances will get reabsorbed. What about water? Water is also getting reabsorbed. So things are moving out. Most of the reabsorption will take place in this particular region. In the descending limb of loop of Henle, there are no transporters. But in the thick ascending limb of loop of Henle, there is sodium potassium 2 chloride symporter. In the early distal convoluted tubules, that is sodium chloride symporter. Okay. Whereas in the descending limb, water can get reabsorbed. Okay, water reabsorption will take place, but no other solutes can enter. Whereas ascending limb is impermeable to water, only sodium potassium 2 chloride symporter is present. In the distal convoluted tubule, early distal convoluted tubule, sodium chloride symporter is present. So sodium and chloride will move into the blood. Whereas the later part of the distal convoluted tubule and the collecting ducts mainly control the amount of water, because here the ADH will act in this particular region and this will mainly regulate what type of urine is formed. Okay, so let us discuss this process of urine formation in detail, formation of the dilute urine first. Okay, so as this particular filtrate is formed in the capsular space, okay, by the filtration of the blood, the glomerular filtrate. Okay, now this particular filtrate, it has the same ratio of water and solute particles as blood. Now why this is important? Okay. So you need to know about the osmolarity before you understand this particular part. So it becomes very clear. So we have already discussed about the osmolarity. We have seen what osmosis is. Okay, so when any particular two areas are separated by a semi-permeable membrane, if this is a semi-permeable membrane, say for example, and if there is a fluid which is present over here which is more concentrated, and if there is a fluid which is less concentrated, okay, here there are more number of solutes. So what happens? What happens to the water? In which direction it will move? There is in the state of non-equilibrium across the semi-permeable membrane, right. So, this is permeable. Say for example, if to the movement of the water, what will happen? Water will move by the process of osmosis, in which direction? From the dilute solution towards the concentrated solution, to maintain the balance. That is, the ratio of solute to the solvent will be balanced. Okay, the osmolarity will be balanced. Now, osmolarity of this solution, osmolarity, it will be high because the solution is concentrated, right. And what about the osmolarity over here? There will be low osmolarity. That's because this particular solution is dilute. Okay, and if there is a difference in the osmolarity, it promotes the movement of the solvent. That is, water can move from the area of dilute solution towards the area of the concentrated solution. Okay. Now, as this filtrate is formed, it has the osmolarity which is about 300 milliosmoles per liter. That is the unit of the osmolarity. Okay. Now, this osmolarity, it is the same as that of the blood. Now, why this is same as that of blood? Now, filtration has taken place, everything does not get filtered out, right, but still the osmolarity is almost the same as that of blood. Now, why? That is because your filtrate is having the same ratio of water and solute particles as that of blood. So, that is why the osmolarity does not change. Even as the fluid is flowing through the proximal convoluted tubules, reabsorption will take place, but has the osmolarity changed? It is the same, right? It is still isotonic to plasma. Why? Because the proportionate amount of water and solute has got reabsorbed in the proximal convoluted tubule. We have seen that a lot of reabsorption takes place, right? So, a lot of sodium mainly will get reabsorbed in this particular area. Apart from that, other substances like bicarbonate, calcium, magnesium, everything will get reabsorbed, but proportionate amount of water is also getting reabsorbed. That is why the osmolarity does not change, it remains the same, even though it leaves the proximal convoluted tubule. Okay. Now, as the fluid is flowing through the next part of the nephron, that is the loop of Henle, that is the next part, right. So, it will enter into the descending limb first. So, it will travel into the medulla, this is your cortex, that is your medulla, and the loop of Henle, as we have seen, that it enters into the medulla, right. It makes a hairpin turn and then it returns back to the cortex, right. So, usually most part of the loop of Henle is located in the medulla, especially with the juxtamedullary nephrons, okay, which have a long loop of Henle. Now, here as the fluid is descending downwards into the medulla, as you can see. So, it is moving into the deeper layers of the medulla. So, as it is travelling along this descending limb of loop of Henle, the osmolarity of the tubular fluid, what is happening to the osmolarity? Here it was 300, now it is 350, it has become 550, still deeper layers 750, 900. So, what has happened to the osmolarity? It is increasing. Further, as it flows through the ascending limb of loop of Henle, as it is ascending upwards, still the osmolarity is decreasing. Okay. So, it is decreasing as it is travelling along the ascending. As it is travelling along the descending limb of loop of Henle, osmolarity is increasing. But what is happening to the osmolarity as it is travelling along the ascending limb of loop of Henle? It is decreasing. As it travels further along the distal convoluted tubule, it is still decreasing, right? It has come down to 100. Okay, now why this is happening? Okay, so that's because of the variable processes of reabsorption and secretions which are taking place as the fluid is traveling along these tubular structure. And the second reason is the osmotic gradient within the interstitial fluid. Now that's your interstitial fluid, right? This is the tubule. There will be cells lining these tubules, and from here we have seen that any substance which is reabsorbed, it will enter into the interstitial fluid. So the osmolarity of this interstitial fluid in the renal medulla, it becomes progressively more. As you can see, this is the osmotic gradient which is maintained in the medulla. So in the layers which are closer to the cortex, it is 300, but as you go deeper, it is increasing, right? 400, 600, 800, 1000, 1200. So the osmolarity is going on increasing. Now this change in the osmolarity of the interstitial fluid in the renal medulla, that makes it possible for more and more water to be reabsorbed by the osmosis. Remember why water moves? Now what has happened? Osmolarity, as you go into the deeper layers of the medulla, what is happening to your osmolarity? Osmolarity is going on increasing. So what will happen to the water now, as osmolarity is more now in the interstitial fluid as compared to the tubular fluids. So in which direction water will move? Now in the descending limb of loop of Henle, is it permeable to any solutes? Is it permeable? No, it is permeable only to water, right? Only water can be reabsorbed. So as the osmolarity is increasing, what will happen to water? Solutes cannot move up or move into the tubules, but it is permeable to water. So what will happen? It will enter into the interstitial fluid. Okay. Now the cells which are lining your ascending limb of loop of Henle, that is however permeable to the solutes, right? Okay. So in the descending limb of loop of Henle, what is getting reabsorbed? Here water is getting reabsorbed. That is the reason why the osmolarity of the tubular fluid is going on increasing, right, from 300 to say 900. But in case of ascending limb of loop of Henle, especially the thick ascending limb of loop of Henle, it has a transporter, right, for the movement of the solute. Sodium potassium 2 chloride symporter is present over here, right? Is it permeable to water? No, it is not permeable to water, but only ions can move out of the tubular fluid. Okay, so from the tubular fluids, the sodium, potassium and chloride, actually potassium is recycled over there through the leakage channels. Okay, mainly only the sodium and chloride will be moving out of the thick ascending limb of loop of Henle. So it will enter into the interstitial fluid, and from here it will enter into the vasa recta. Remember vasa recta, these are the blood vessels which are supplying to the loop of Henle, right? It is peritubular capillaries also and vasa recta also. So from interstitial fluid it will move into the vasa recta. Remember these vasa recta which are present over here. Okay, so it will move into the blood capillaries. Now as these fluids are passing along the ascending limb of loop of Henle, we have seen that due to the presence of sodium potassium 2 chloride symporter, ions are moving into the interstitial fluid. But what about the water permeability? It is quite low, right? So water is not reabsorbed over here. What is getting reabsorbed? The ions are getting reabsorbed, right? Water is not getting reabsorbed. So what will happen to the osmolarity? Here ions are moving, water is not moving, water is remaining over here. H2O remains over here. So now what will happen to your osmolarity? It was high, but as it is moving along the thick ascending limb of loop of Henle, ions are moving out, but water remains over here in the tubular fluid. So what will happen to the osmolarity? Since there is high amount of water, high amount of solvent, osmolarity will go on decreasing as it is traveling along the thick ascending limb of loop of Henle. So as it leaves the loop of Henle, the osmolarity of the tubular fluid, it drops down to 150. Okay, so it becomes round about 150 milliosmoles per liter. So the fluid which is entering into the distal convoluted tubules, this is much more dilute than the plasma, right? What is the osmolarity of plasma? It is 300, right? And now it has become almost the half, that is 150. Now as the fluid continues to flow along this distal convoluted tubules, again here additional solutes will be reabsorbed, right? What type of transporter is present in the early DCT? It is the sodium chloride symporter which is present over here. So sodium and chloride will be reabsorbed. Now again the solutes are moving into the interstitial fluid over here, and only few water molecules are reabsorbed. So what will happen now? Osmolarity of this fluid as it is traveling along the distal convoluted tubule, it will drop further, right? Now here you can see it is 100 milliosmoles per liter. Now from here the fluid will enter into the late DCT and the collecting ducts. Now the osmolarity of the fluid which is entering over here, it is 100. Now as it flows along the late part of the distal convoluted tubules or the collecting ducts, now here the types of cells which are lining the tubular wall, these are sensitive to the levels of ADH, right? So when very low levels of ADH are present, if the ADH levels are low, what will happen? These are the principal cells, right, into which the vesicles will be present which contain the aquaporine channels. So when ADH level is low, what happens to those aquaporine channels? Remember the aquaporine channels which are inserted, when the ADH levels are low, these aquaporine channels are removed from here and they will enter back inside the cells. So what will happen to the water reabsorption in this particular region then? In case when the ADH levels are low, aquaporine channels will be removed, so water reabsorption will be decreased. So as a result of decrease in the reabsorption of water as it is traveling along the collecting ducts, water reabsorption of your H2O, it is decreased. So what will happen? Water will remain over here, right? So more amount of water will remain in your collecting ducts. So as a result of this, what will happen to the osmolarity of the urine? It was 100 as it is entering over here. Now it has reduced to 90. Okay. Still further 80, 70, 65. Why it is happening? That's because the water reabsorption is getting reduced. Okay. So by the time the urine it leaves the collecting ducts, the osmolarity is round about 65 to 70 milliosmoles per liter. That is 4 times more dilute as compared to the blood plasma or the glomerular filtrate, you can say. What was it? In the blood or the glomerular filtrate, what was the osmolarity? Yes, it was 300, right? But now how much it has become? Round about 65. So it is 4 times more dilute now. The urine which is produced, it is a dilute urine. Why? Because the osmolarity is very low, right? Because the amount of water which is excreted in the urine is very high. That's why the osmolarity is low. That means dilute urine is produced. Now what makes it possible? Yes, the very low levels of the ADH makes it possible for the dilute urine to be formed. So it is to do with the formation of the dilute urine. Now coming to the formation of the concentrated urine. Now when it is formed? When the intake of water is low or water loss is high, say for example heavy sweating, okay, or say for example in case of dehydration or excessive loss of water from the body or fluids from the body, hemorrhages for example. So what the kidneys will do? They will try to maintain the homeostasis of the body fluid. So they will try to conserve more and more water into the body while still eliminating the waste and excess ions which are not required by the body. So that process should continue, right? Removal of the waste. Total urine formation shutdown will not take place, but the concentrated urine will be formed in these particular situations. Okay. Now what makes it possible again? That's because of the ADH. So it's because of the high levels of ADH that the kidneys will produce a small volume of highly concentrated urine. Okay, so now in this particular situation the urine can be 4 times more concentrated as compared to the blood plasma or the glomerular filtrate. So it is 300 milliosmoles per liter, right? So the urine which is formed, it can be having the osmolarity of up to 1200 milliosmoles per liter. So it is 4 times more concentrated than the blood plasma or the glomerular filtrate. Okay. Now actually we have seen that the ADH is responsible for the production of the concentrated urine, right? But it is aided by the presence of the osmotic gradient of the solutes in the interstitial fluid. That is very important. That's the interstitial fluid, right, of the medulla, and this is the interstitial fluid in the renal cortex. So alone the ADH cannot make it possible for the formation of the concentrated urine. So it must be aided by the differential osmotic gradient which is present in the interstitium. Okay, so as we have seen that the osmolarity of the interstitial fluid, it increases from 300 as you move deeper into the medulla. It may increase from 300 to round about 1200 in the deeper layers of the renal medulla. So what makes it possible to maintain this osmotic gradient? It should be maintained, right? It should not get disrupted. So it's because of the movement of these 3 substances, sodium, chloride and urea, which makes it possible for the building up and maintenance of this osmotic gradient. So what makes it possible? That's the differences in the solute and water permeability. As the fluid is traveling along the tubules, there is a difference in the movement of the solutes of water out of this tubular fluid, right? It is variable depending upon the different parts of the nephrons. Okay, so this difference in the movement of the solute and water in either directions, may it be the secretion or the reabsorption. So it is variable. That makes it possible for maintenance of this osmotic gradient. Another reason why it is made possible is because of the countercurrent flow of the fluids into the tubular structures in the renal medulla. Now what do you mean by this countercurrent flow? Countercurrent flow means flow of the fluid in the opposite directions. Now when this occurs, when 2 tubes run in the opposite direction, okay, counter to each other, the fluid flowing through these 2 tubules must run in opposite direction. Now does it happen anywhere in the nephrons? Yes. Okay, so there are two locations where this counter current flow is taking place. The first one is the loop of Henle. Now how does the fluid travels? In the descending limb of loop of Henle, it's in this direction. And what about the ascending limb of loop of Henle? It's in this direction, right? So fluid is moving in opposite direction. So that's a counter current flow. What is the second place where it happens? Yes, that is in the vasa recta, which is supplying, right? These are your vasa recta, which is supplying the loop of Henle. Now in which direction the blood is moving? In the descending part of the vasa recta, the blood is moving in this direction. And in the ascending part, in which direction blood is moving? It's in the opposite direction. So that's again a counter current flow, right? So these counter current flow makes it possible for this osmotic gradient to be maintained. Okay, now there are two mechanisms which are responsible for this maintenance of the osmotic gradient, build up of osmotic gradient and the maintenance of the osmotic gradient. That is the counter current multiplication and the counter current exchange. Okay, let us talk about the counter current multiplication. Now what is this counter current multiplication? Now, actually the build up of the osmotic gradient, as you travel deeper into the medulla, the osmotic gradient increases. That is made possible by this counter current multiplication. So what is this counter current multiplication? It is a process by which a progressively increasing osmotic gradient, it is formed in the interstitial fluid of the renal medulla as a result of counter current flow. So as you travel deeper into the medulla, there is build up of the osmotic gradient, there is increase in the osmotic gradient, right? It is not the same. Osmolarity increases as you go into the deeper layers of the renal medulla. So this progressive increase in the osmolarity in the renal medulla as a result of counter current flow, that is called as the counter current multiplication. Okay, so it is resulting in the multiplication or increase in the osmotic gradient. Okay, so what makes it possible? This counter current multiplication, it involves the long loops of Henle in the juxtamedullary nephrons. Okay, so here you can see that there is a counter current flow, right? So fluids are moving in the opposite direction. So since this counter current flow is helping in establishing this osmotic gradient in the renal medulla, the long loop of Henle, where it is happening, that's because of the flow of fluid in the long loop of Henle. That's why these long loop of Henle, these function as the counter current multiplier. These are also known as counter current multiplier, because they are responsible for that counter current multiplication, which is responsible for the build up of this osmotic gradient. Okay, so this osmotic gradient, variation in the osmotic gradient is utilized by the kidneys to produce the concentrated urine. Okay, so how this concentrated urine is produced? Let's talk about the formation of the concentrated urine step by step. Okay, so first we will discuss the counter current multiplication. Okay, now what makes it possible? That's because of the differential movement of the solutes and the water at the different parts of the loop of Henle, right? Now there are symporters which are present in the thick ascending loop of Henle, correct? So they will reabsorb the sodium and chloride, right? Potassium is just circulated over there. Okay, so actually the net reabsorption of sodium and chloride will take place into the blood. Okay, just to balance it, actually the potassium also is reabsorbed from the tubules into the cells, but they are secreted back into the tubules. Okay, so in this part the reabsorption of the sodium and the chloride ions will take place. So these ions, they will start entering into the interstitial fluid. So there will be a build-up of these ions in the renal medulla, right, due to the presence of this transporter, sodium potassium 2 chloride symporter. Okay, now in this particular region, as we have seen that water is not reabsorbed because the cells are impermeable to water, right. So as a result of this, what will happen? The sodium and chloride, there will be a build-up of these ions in the interstitial fluid. Okay, so only the ions are moving out of the tubular fluids into the interstitial fluid. Okay, so the osmolarity is increasing over here due to the build-up of the sodium and the chloride in the interstitial fluid. Now there is a countercurrent flow, right, through the descending and the ascending limb of loop of Henle. So this actually helps in establishing the osmotic gradient over here. Now what makes it possible for this sodium and chloride to get reabsorbed over here? Osmotic gradient is maintained because of this reabsorption. Now what makes it possible? Now supposing if there was no countercurrent flow, what would have happened? From where would have these sodium and chloride come over here? Actually it is present over here, right, in the tubule. Now because of this countercurrent flow, what is happening? The concentrated solution is entering into the ascending limb of loop of Henle, right. So imagine if the osmolarity was not this much, what would have happened? The reabsorption of the ions would not have taken place, right. So this countercurrent flow of the fluid makes it possible to provide the supply of these ions for it to get reabsorbed in this particular region to maintain the osmolarity of your interstitial fluid, right. Okay, so as a result of this continuous supply of the sodium and chloride over here, more and more sodium chloride will be reabsorbed and it will help in the build-up of the osmolarity in the interstitial fluid of the medulla. So this differential reabsorption of these ions makes it possible for this gradient to be formed in this particular region. Now from where this concentrated solution will come into the ascending limb of loop of Henle, what makes it possible? Usually what is the osmolarity? It is just 300, right. Now how it becomes 1200? That's because the descending limb of loop of Henle, it is impermeable to any solutes. No solutes are getting reabsorbed over here, right. But what it is permeable to? It is permeable to water. So what will happen? Water is getting reabsorbed over here, right, in the descending limb of loop of Henle. Now why this water is getting reabsorbed? There should be some reason, right. When did I tell you that water will move by the process of osmosis? When there is a difference in concentration across the membrane, right. So because the interstitial fluid in the medulla, it is rich in sodium and chloride, right. So what has happened to the osmolarity of the interstitial fluid? It is high, and the osmolarity of the fluid which is present over here, that is low. So in which direction water will move? What did I tell you? This is a semi-permeable membrane. So water will move from the fluid which is having low osmolarity to the fluid which has high osmolarity. This is the reason why water is getting reabsorbed into the interstitial fluid. Now as a result of this, what is happening? The osmolarity of the tubular fluid is increasing, right. Here it was 300, it is now increased to 380, 580, 780, 980 and then to 1200. Okay. So this countercurrent flow makes it possible for this maintenance of the gradient or setup of the gradient. Remember, if water reabsorption does not take place, what will happen? Osmolarity will not change. If this osmolarity is not high, reabsorption of the ions also will not take place. So from where this gradient build-up will take place? It is not possible. Okay, so this countercurrent flow of the fluids, it makes it possible for the change in the osmolarity in the interstitial fluid as well as change in the osmolarity in the tubular fluids. So as you can see in the descending limb of loop of Henle, the osmolarity is going on increasing, and as it ascends it is going on decreasing. That's because of the differential permeability to water and the solutes and the countercurrent flow of the tubular fluid. Okay, so this is why the osmolarity of the fluids is going on reducing. So as it leaves the loop of Henle, the osmolarity of the fluid is as low as 100 milliosmoles per liter at the junction of the medulla and the cortex. So as it is entering the distal convoluted tubule, the osmolarity is very low. Now, in the collecting duct, so as it flows along, as the fluid enters into the collecting ducts, now here there will be influence of the ADH, right? Now, as the ADH levels are high, what will happen to the water permeability in this particular region? It will be increased, right, of the principal cells, because aquaporine channels will be introduced as a result of high levels of ADH. So what will happen to the reabsorption of water? As you can see that more and more water is getting reabsorbed into the interstitial fluid, right, by the process of osmosis. So from here the water will enter into the vasa recta. So more and more water is getting reabsorbed in the collecting ducts. So as a result of reabsorption of water, what will happen to the fluid which is present in the tubules? As you can see, it was 300. What is happening to your osmolarity? It is increasing, right, as more and more water is reabsorbed. It is 320, then it has become 400, 600, 800, 1000, 1200. So osmolarity is going on increasing because water is leaving. Is there any solute which is leaving this particular region? Are there any transporters in this region? No, it is mainly the water which is leaving in these later parts of the collecting ducts. Okay, because there are two different types of cells and mainly these are responsible for the reabsorption of the water under the influence of the ADH. So as more and more water is leaving the fluid, the osmolarity of the fluid which is travelling, it is going on increasing. Okay, because the solute concentration will go on increasing. Now urea is one of the important component which is present in the fluids over here. So as water is leaving the tubules, the amount of urea in the tubular fluid is going on increasing. Okay, so the solution will become more and more concentrated. But in the deeper layers of the medulla, what you can see over here, these later parts of the collecting ducts, these are permeable to the urea. These portions are impermeable, the starting part, but the later part is permeable to the urea. So what will happen? Now here the osmolarity is very high. So what will happen? The urea, it will move out of the tubular fluid into the interstitial fluid. So as the urea leaves out, what happens to this urea? Where it will go? Does it go to the blood? No, it doesn't happen like that. It is not reabsorbed. Actually the urea which is leaving the tubular fluid, it is recycled. Now what do you mean by recycling? Something which is released out and then it is brought back. So that is recycling, right? So this urea recycling it actually is responsible for this buildup of urea in the renal medulla. Okay, so this process of recycling, let us understand what is this process of recycling of the urea. So as we have seen that the later parts of the collecting ducts, these are permeable to urea, so it will enter into the interstitial fluid. Now from here, this urea, where it is going, it is entering back into the tubules, right? As you can see that the descending limb of loop of Henle, the later deeper areas, it is permeable to the urea. Even the thin ascending limb of loop of Henle, it is also permeable to urea. So what is happening to this urea which is there in the interstitial fluid? Yes, it is entering back into the tubules, right, which was reabsorbed over here in the later parts of the collecting ducts. It is entering back into the tubules. Okay, so as the fluid is flowing now urea has returned back to the tubular fluids. So now as the fluid is traveling, are these areas reabsorbing the urea? In any of these regions we have discussed, is it permeable to urea? No. The thick ascending limb of loop of Henle and the distal convoluted tubules, it is impermeable to urea. So urea will remain in the tubular fluid. So as a result of this and as a result of reabsorption of water, the osmolarity, what is happening to the osmolarity of the tubular fluid? It is going on increasing. So what it is helping in? It is helping in the increase in the osmolarity over here. Now again the osmolarity will increase, but as it reaches the later part of the collecting ducts, what will happen? Again it will be reabsorbed into the interstitial fluid, right? From here where it will go? It will go back into the tubules, travel along these tubular structures and reach back again over here. So what is this process now? Here the urea, it is getting recycled, right? It is getting exchanged in between the tubules. So it is just recycling over here. So this particular constant transfer of this urea in between these segments, that is known as the urea recycling. So again this urea also is helping in this maintenance of the interstitial osmotic gradient and the reabsorption of water also. Okay, so reabsorption of the water will be promoted because of the solute content is very high over here. Water will get reabsorbed into the interstitial fluid in the renal medulla. So as a result of this, what will happen? Urine will become more and more concentrated. So concentrated urine will be excreted. Okay, so this is how the countercurrent multiplication will help in maintaining the osmotic gradient, and that's how the concentrated urine will be formed. So it's not alone ADH, but it is the alteration in the osmotic gradient also which is responsible for the formation of the concentrated urine. Now there is one more process which is involved over here, that is countercurrent exchange. Now what is this countercurrent exchange? That is a process by which solute and the water, it passively is exchanged between the blood which is present in the vasa recta, that is over here, and the interstitial fluid which is present in the renal medulla, because of the countercurrent flow which is present over here. Okay, so this vasa recta also contains the descending part and the ascending part, right? So there is a countercurrent flow of the blood. So actually this countercurrent flow makes it possible for the exchange of substances in between the blood and the interstitial fluid. Now let us see what is the importance of this countercurrent exchange. Okay, so as we have seen in case of loop of Henle, there is a countercurrent flow, right, of the fluid, tubular fluid. In the same way the blood is also flowing in the opposite direction. So there is a countercurrent flow. So this countercurrent flow makes it possible for the substances to get exchanged in between the blood into the vasa recta and the interstitial fluid. So that's why vasa recta is said to be functioning as the countercurrent exchanger, because this exchange of substances is taking place in the vasa recta. Now let us see the osmolarity of the blood which is entering this vasa recta. So osmolarity, it is 300, right? 300 milliosmoles per liter. So same as the fluid which is entering the loop of Henle, right? Now as the blood flows along the descending parts of the vasa recta, so it is entering the region wherein a lot of sodium chloride ions and the urea will be present, right? Remember this, osmotic gradient is being maintained by the countercurrent multiplication, right, as we have seen just now. So because of this osmotic gradient, as the blood is flowing along this descending part of the vasa recta, it is entering into the area where the osmolarity is very high. So the concentration of these ions is very high in this region, the interstitial region. We have seen that reabsorption takes place, right? So very high level of these ions are present over here. Even urea is present. So as the blood is descending downwards, it is entering the area wherein the interstitial fluid has a very high osmolarity. So as a result of this, what will happen now? Concentration gradients are in which direction? Sodium and chloride ions are present in the interstitial fluid. Less amount will be in the blood. So in which direction it will flow? Yes, of course it will enter into the blood. What about the blood, water which is present in the blood? In which direction it will flow? Osmolarity of the blood is low. Osmolarity of the interstitial fluid is high. So what will happen to the movement of water? Yes, it will leave the blood, right? So it will move out of the blood as the fluids descend down, or the blood descends downwards. So what has happened to the osmolarity of blood? As you can see, it is 500 over here, 700, 900, 1100, 1200. Why it has happened? That's because sodium and chloride is entering. Solutes are entering, water is leaving. So what will happen to the osmolarity? Obviously it is becoming more and more concentrated. So osmolarity will increase. Okay. But as it enters the ascending part, now in which direction blood is moving? In the opposite direction, right? So in the region where there was very high osmolarity, it is now traveling towards a region wherein the osmolarity is very low, right? It is 300. That is flowing in the opposite direction. Now, as a result of this, what will happen now? Osmolarity is going on decreasing as the blood is traveling towards the cortex. So, as the gradient, osmotic gradient is going on reducing, what will happen to this fluid or the blood? Will it travel as it is? No. What is happening to the ions now? Sodium and chloride ions are moving out because the concentration gradient is in the opposite direction now. Because of the reabsorption, the gradients have reversed. So as the blood is leaving the ascending part, now the sodium and chloride will move out of the blood. And what will happen to the water now? The osmolarity of the blood is very high as compared to the interstitial fluid. So in which direction it will move? Obviously, it will move from interstitial fluid into the blood, right? So as a result of this, solute is moving out, water is entering the blood. So what will happen to the osmolarity of the blood? From 1200 it has increased to 1100, 800, 600, 400. Osmolarity of the blood is going on decreasing. Okay, so as you can see, the osmolarity of the blood which is leaving the vasa recta, it is slightly high, right? It is 300, but it is just 320. So there is a slight increase in osmolarity. It does not change. So what is the role of this countercurrent exchange? What is the role? There is an exchange of substances in between the interstitial fluid and the blood. What is the role over here? So it provides the oxygen and the nutrients to the renal medulla. But as it is moving out of the medulla, is it disrupting the osmotic gradient which was actually developed by this loop of Henle? Is it washing out or diminishing the osmotic gradient? Is it getting disrupted? The ions which were entering into the blood, it has left the blood before the blood leaves the medulla. Water which is moved out, the water, same amount of water almost, it has entered the blood back again. So has it disrupted the osmotic gradient? Ionic concentration did not change. Water concentration did not change in the interstitial fluid. So this osmotic gradient, it is not disrupted by this countercurrent exchange. So it will help in providing the oxygen and nutrients without disrupting this osmotic gradient. Okay, so what you can conclude from this? The long loop of Henle, it establishes the osmotic gradient in the renal medulla by the countercurrent multiplication. And the vasa recta, it helps in maintaining the osmotic gradient in the renal medulla by countercurrent exchange. Okay, so the one countercurrent flow, it is building up the osmotic gradient, which makes it possible for the reabsorption of various substances as the fluid is traveling along these tubules. And the another one, that is a countercurrent exchange, it helps in maintaining the osmotic gradient in the medulla. Okay, so this is how the concentrated urine will be formed by the kidneys, okay, in the presence of high levels of the ADH. But you need to understand what makes it possible. It's because of these countercurrent multiplication and countercurrent exchange which makes it possible for the concentrated urine to be formed. Okay, so this is to do with the formation of the dilute and concentrated urine, that is the physiology of urine formation. This is a reference. Thank you for watching.